I want to talk about discrete characters. Now you may say, well, this whole course is about continuous characters. What are you doing? You're wasting time. But it will be a discrete character model that involves the evolution of continuous underlying characters. And I want to first talk about a little bit about how people have treated discrete characters. Now, discrete characters can include simple 0, 1 type traits, which there are two states, red and green or whatever, and you model them maybe by calling 1, 0 and 1, 1. But they can get more complex than that. And you can have multiple states, and one of the ways is to have the multiple states be on a linear scale, so you have number of scales in a garter snake, and they're discrete values, but they're linearly ordered. We were carefully taught in graduate school. There's a name for that, and everyone's forgotten the name. I think you forgot it too. Meristic traits, right? M-E-R-I-S-T-I-C. But now that you've said meristic, you must know something, right? Okay. And I'll get a little bit back to that. I'm going to just spend my time on two state characters because that's what we've got a preliminary model for. And this is some work. Well, I'll explain. It's work that actually goes back a long way, and starts with a famous person. Most people who are faced with meristic character, with not meristic characters, with discrete characters, will, in phylogeny analysis, they will use parsimony methods for them. And although there have been big wars, philosophical political wars in systematics among morphologists, and whether parsimony is philosophically mandated or whether, and therefore true, or whether statistical approaches are as good or better. Although there have been those wars, most morphologists out there, if you press them, say, why are you using parsimony? They'll say, I don't have any other method. I mean, nobody's presented me with another approach. There used to be a lot of discussion among systematists, morphologically inclined systematists, about something called the character coding problem. It also was the case that people would take traits that were continuous and not really wanting to treat them continuously, they would code them into large and small. And there was the character or curved and straight. And there was what was called the character coding problem, which was how to do that, how to take a continuous trait and reduce it to discrete. There even came to be, among people using parsimony methods in the 80s, a bit of lore that circulated saying that if you wanted to do parsimony, you had to have discrete characters. And so if they started out continuous, you had to somehow convert them. Actually, some early parsimony methods didn't use discrete states. But anyway, people would trouble themselves about the character coding problem. There's really no statistical model underlying parsimony, although if you're willing to assume that the traits are independent of each other, which is a very strong assumption, then you could do bootstrapping or jackknifing, so there is to that extent some statistics you could use. I have argued that when faced with continuous traits and turning them into discrete traits, a good attitude is just say no. Leave them as continuous traits and do all the other stuff we've been talking about. Now, if you have to have a discrete character, if instead of a nice continuous scale you have something that is self-evidently discrete, then what do? There are statistical models for them. The one that's gained some traction was already mentioned by Brian, I think. Was it? Yeah. And that's Mark Pagel's and Paul Lewis's use of something that's now called the MK model. You were introduced to this. Mark Pagel is at the University of Reading in England. He's very well known now for his work on statistical phylogenetic approaches to language evolution. Many people think of him as an Englishman. He's actually an American who did his PhD at the University of Washington, although in another department, so I didn't meet him until he was departing. And he was co-author with Paul Harvey on a very important book on comparative methods in 1991. Paul Lewis, well, Pagel's interest in this was to have two or more such traits and then put forward a likelihood framework in which you tested a model in which the characters evolved in a covarying fashion versus them evolving separately. And that was a pretty good breakthrough in 1994. Paul Lewis picked up the model and said, let's use it to infer phylogenies for morphological traits, and he added in some wrinkles about enabling you to have different rates at different characters and drawn from a prior distribution of rates. So they've both done interesting things. And their model is one in which you basically start out knowing the nucleotide models of statistical molecular phylogenetics. And you say, well, let's just make it a nucleotide. There's this DNA site with only two nucleotides at it, and we have rates of change forward and backward between them. And of course, then the statistical machinery is all is available. The statistical work of figuring out transition probabilities and building programs more or less done at that point. So they're treating them as states from discrete states that simply pop back and forth between each other, and that each little slice of time dt, there's a probability alpha dt, a small probability. If you have a 0, that it will change to a 1. There's also a probability beta dt. If you have a 1, there's a probability beta dt that it will, in that interval, change to a 0. Okay. They're not multiple locus models. They're not saying what the genetics is. And before, whoops, we'll get there. Before we say that, I'll mention this in a moment again. They don't talk about variation within the species. They just assume the species characterized by 0 or characterized by 1, and then there's no process talked about of how you get from 0 to 1 or what its underlying genetic basis might be. The alternative to this actually already existed and had been in existence for 60 years. And that is the threshold model, which was invented by this fellow who you may have observed. You may know about, and that's Sewell Wright, who not only was one of the two most important of the three great founders of theoretical population genetics, thereby I have a population geneticist prejudice that those people, Fisher, Wright, and Haldane, were who really kicked off the modern synthesis. And he had the great advantage over Haldane and Fisher of living longer. So he got to win all the arguments by being around. And he also had the advantage that because he lived until 1998, he died in his 99th year with a paper in press. A lot of us older folk in the field knew him. I knew him. Steve, you met him? Yep. Sure. Everybody met Sewell Wright. Everyone over a certain age. Okay. Here he is shown in 1955, which is the occasion on which he retired at the University of Chicago, then was moved to University of Wisconsin in Madison, which let him be an active faculty for another 5 years, and he retired a second time. He did not only theoretical population genetics, he founded path coefficients, did one of the first graphical models, all sorts of things. Oh, and he was an important figure in American mammalian genetics and working on interaction of genes with coat color. He, for example, wrote a series of review articles in 1917 on genetics of coat colors and concluded that the best way of explaining coat color interactions, he had reviewed it for a number of species, was that there were genes controlling enzymes. And this was a predecessor of one gene, one enzyme. And in fact, in the one gene, one enzyme, he then wrote review articles on that topic every 5 years thereafter. And in 1939, when Beadle and Tatum did the great one gene, one enzyme paper, they cite Sewell Wright's review articles. They knew all about that. Well, he did years of work. His organism was the guinea pig. And he did years of work on guinea pigs. And he would bring them into class for demonstration. So here he is showing in his photograph, which is also his photograph for being inducted into the National Academy of Sciences. He wrote his diffusion equations on the blackboard and posed with a guinea pig. And there's been a persistent rumor that he then turned around absent-mindedly and started to erase the board with the guinea pig. He was the sort of ultimate absent-minded professor, not a person who was easy to make small talk with. He was always mumbling about, well, in my 1934 paper I said this, in my 1942 paper I said that. And he was one of the world's worst lecturers. Have you heard him lecture? Would you agree?
I can think of worse lecturers.
Oh, okay. Yeah, he's pretty bad. He's fine. And what he would do is he would be at the board, and he would be facing the board, mumbling and drawing equations on the board. He often had an eraser in the other hand, and as he went along, he would erase what he wrote. And he would typically dress like that, and usually a three-piece suit with a vest. And he would put the eraser under his arm. And so when he was doing this, he could always have access to the eraser. Okay. Sewell Wright always furiously denied that he had erased the board with a guinea pig. He couldn't stand this story. Other people who had taken courses from Wright at the University of Chicago said, oh, they'd seen him do it. And there is he would bring guinea pigs into class for demonstrations. I have here a guinea pig. Well, okay, it's not a living guinea pig. It's a toy. It's a stuffed guinea pig, courtesy of Brian O'Meara, who did something to it that I'll tell you about in a moment. Now, he'd have the guinea pig. He'd have the guinea pig on here, and they were pretty placid animals, and he'd put them down, and they'd wander around. He'd say, "Look at the coat color," and he'd go back to talking about whatever else, and the guinea pig be wandering. And people said they'd seen cases where the guinea pig started to go too far. And now if you've worked with rabbits or guinea pigs, and I have been a technician with rabbits between high school and college for a few months. I didn't like rabbits, they didn't like me. But the one thing about them is they live in holes in nature, and so do guinea pigs. And the way you calm a rabbit down, I found out, I had to, was that you take it, you put it under your arm, and you cover its eyes with your arm and your body. Anybody else here work on mammals like that and know, am I wrong? Is this right? You cover them, you put your arm over one eye and your body over the other, and then the animal, being really brilliant, says, oh, I'm in a hole, I'm happy, and it relaxes. Okay. So Sewell Wright is up there, and the guinea pig starts to escape, and so he puts it under his arm and goes back to writing on the board, and of course then the inevitable happens. That's where he keeps the eraser, okay? So he's got, he's got his diffusion equation up there, and he needs to erase something, and so inevitably, now this worked because Brian O'Meara put an eraser on the bottom of this guinea pig, and I thank him for donating this to my lecturing props repertoire. Okay. So Sewell Wright had all this work on guinea pigs, and in the process, he had a model, he had a trait which was the number of toes on the hind limb of the guinea pig, and it was either 3 or 4. And some strains had mostly 3, and some had mostly 4. So he made the following model. And published it in 1934. For the result of crosses between different lines of guinea pigs and the number of toes that they had. He said, well, let us suppose there is some underlying quantity, a character, that we can't observe. It's come to be called in that literature the liability. Okay, but liability is a hidden state. We can't observe it. But it's polygenic, and it's controlled by many genes, presumably, It's something out there in the developmental process. And it varies. And some lines are, each line has its distribution of amount of it. And then there is a threshold. Now in Wright's case, there are actually two thresholds. It was a little more complicated. But in the simple threshold model, there's a threshold. And if you were to the right, if the amount of liability is above the threshold, you develop 4 toes, and below the threshold, you develop 3 toes. So that's the model. And it's a polygenic model. It's got this hidden state. It's very different from the 0-1 model, the MK model. He used it to predict results of crosses among different lines with apparent success in 1934. Well, we can take that model, just simply use for the liability, just treat it like the traits we're working with here, and we can say, well, okay, let's let the liability evolve by something like Brownian motion or Ornstein-Uhlenbeck, whatever you want. Here we're just cartooning that model, and as the mean of the population shifts, you get a greater fraction of individuals with 4 toes versus 3 toes. Now, the model, you can immediately see, has built into it within species variation, within population variation. It's a natural part of the model. Here is a simulation of such a model where we start with a mean right on the threshold. And in that case, if you look at this bar here, we color it in according to the percentage, according to the area to the right, which is exactly 50% here. And I did a simulation and with Brownian motion up this tree, and you can see the fraction of black increases nearly to 1, comes down, increases again. And what I put at the nodes is the phenotypic, the mean liability in standard deviation units. So here you've gone up to 4 standard deviations, you're way off to the right. Here you drift back, you wander back down by Brownian motion to 1.8, and you start to see a few zeros, a few white ones instead of black, wandering back up here, here wandering down, having almost no shaded individuals, dark individuals, minus 2 standard deviations, wandering back up in this line. So that's the kind of evolution you'd expect to see in this model. Now, it has some advantages, some great advantages. If the population crosses the threshold, soon afterwards, it is much more likely soon afterwards that we will come back. But if you wait longer, if there's time for it to wander farther from the threshold, get way beyond it, then it's much less likely in the next little period that you will wander back over the threshold. That has some feel of biological reasonable realism to me. Okay. The MK model never does that. Once you arrive at a state, your chance is the same in every little instant thereafter of coming back. It doesn't have that. Brian referred to this when he talked about changing rates. It's a little bit hard to think of this in terms of rates, but probabilities of change, yeah. It predicts a phase of polymorphism. If you move from having all white to all black, you go through a region of polymorphism, and you expect that. So, and then it naturally connects to within species to analyzing within species variation. But a very important feature is that if you take in the MK model, if you say to yourself, oh, let's put some correlations in between different of these characters, well, you will find yourself proliferating parameters enormously, because you'll be saying, well, for each combination of states at the other characters, what's the chance of change? And I have to have a separate parameter for every such situation. The number of parameters blows up as something like 2 to the number of characters. It's really horrible, and most of them you'd never be able to analyze separately. Here, the underlying model is just a phenotypic it's just phenotypic variation on continuous scales. So it has means and it has covariances, has additive genetic and phenotypic covariance, and that's all. It doesn't have all those 2 to the n parameters. It is more like n squared parameters, n squared over 2, for the covariances. So it says it can handle covariance between characters with a modest number of parameters, as all continuous phenotypic models using normal distributions can. Okay. If you want to compute a likelihood, it's a little tough under this model. It's easy, not too hard under the MK. Because, and here's a diagram just showing you maybe two individuals with one trait, one liability, and imagining, say, your normal distribution is sitting here, and you happen to observe state 1 for the first individual and state 0 for the second individual. Well, the likelihood will be the probability under this part of the distribution. In other words, you take the dome of the bivariate normal distribution, and its total area, total volume under it is 1. How much volume is there under the part? Oh, I'm sorry. The part that is on the appropriate side of each threshold. More formally, here's a case with like 5 individuals connected on a tree. I haven't specified the tree, but it's there. The probability of a particular combination of states is just a multiple integral 5 times, going from either the threshold upwards or from the minus infinity to the threshold, depending on which of these we're talking about. And that's the multivariate normal distribution. So yeah, you have to compute that multiple integral. How do you do it? There's no analytical solution. There are approximate polynomially ways of doing it. They're a pain, but in principle you could do them. It takes a lot of fussing. So what I've done in the program ThreshML, which is in distribution, it's in advance of its release with a new version of Phylip, but so knowing that I was publishing the paper on this, I did put it up at the site, and it's going to be part of our exercise implicitly. I have a program to infer, in this case it's the covariance matrix of evolutionary changes, the evolutionary covariances, using a Markov chain Monte Carlo technique. And what that is I hope you have at least a vague familiarity with what Markov chain Monte Carlo is. We have not spent most of our time here talking about all the fun technical details. What I assume is that somehow we might imagine knowing the liability values at the tips and at the interior nodes. But what we want to do is draw a very large random sample of liability values that fit the observed phenotypes here just for one character. And what we do is we update them. And there are two update processes. For the interior nodes, there's what's called a Gibbs sampler, in which you, oh, I'm sorry, I keep hitting the wrong button. You want to update this, you can look at its immediate neighbors on the tree and the branch lengths, and there is then a nice simple draw from appropriate normal distribution, and you can draw another plausible value for this and update this liability. You keep drawing new ones. You can go around the tree updating the interior ones. When you get to the tips, you do the same thing. You look at only one neighbor, but you also do something else. Anything you draw, you make sure it falls on the correct side of the threshold to account for the phenotype you observe. That is then a metropolis for those involved in the technicalities. It's a metropolis Hastings algorithm, you have to just do that carefully. So I can go around and do these liability updates and draw a very large sample of collections of liabilities. Here is a simulation. This is actually correctly, they're sampled from a run, from a few steps of a run. So here we have the threshold, the dotted line. We have 2 tips that have state 0. We have 5 tips that have state 1. We start out with some liability values for them, and we assign some liability values down in the tree, and then you start going around and reconsidering them one at a time. Here, let me go back. This was here. This one is going to change. We're reconsidering a tip liability, and then we do it for another tip and another. Now, let me go back. Oh, that's not good. That tip managed to move across the threshold. It's not supposed to have done that. You know, I've given this talk many times. Nobody ever called me on it before. Okay, a bad simulation. Okay. And you can imagine updating the tips, and you also go into the interior node. So there is this one, this node here. We're keeping it at the same level, same height, but it's moving sideways on the liability scale. And you keep going. And you can do this. And even at the root. What I actually did, there was that one that went across the threshold. I should redo that. Also, in doing all this, I actually left out one of the nodes. But anyway, you get the idea. The program will draw a big sample. It'll run for a full-size run. Will take about an hour. The runs you will do in your exercise are briefer. And then you can ask about the covariances of change along the tree for each set of liability values, and you can they give very, very simple estimates. If you pretend that you observe the character values at both ends of a branch, and you do this for many different values, you can accumulate that information and simply measure the covariances directly, and you get a very straightforward estimate of the covariance matrix. Tyler.
Hey, actually, I can't see the error that you're pointing at.
Well, maybe there was no error. Maybe I'm... Well...
Could you go to the first one?
Wait. I'll roll it back to the beginning. There are 2 on the left. Okay. Right. This one was a change from who to who. Yeah, it was from here to here. Okay, so that's fine. The next one, yeah, is a change from there to there. It's on the correct side of the threshold. You're right. It was just a change. That's why nobody called me on it before. Because it wasn't wrong. This was on the right side, is still on the right side. I think you're right. There isn't a mistake there. A relief. There's one other step missing, however, which I noticed the other day. Okay, so the program was sampling away. You can you can then come up with an estimate of the covariances. We're actually not getting the likelihoods because we're not reconsidering the tree in this case. We assume the molecules have given us the tree. And we end up with an estimate of the covariance matrix between the characters, and that's of interest because you want to know in which directions are things changing, what are the principal components of the covariance matrix, for example, what's an estimate of it. There's all sorts of things of interest. Here's a computer simulation of 2 characters evolving. I don't remember all the parameters. There are 50 tips. They're just labeled with letters or letter pairs. And the tree is generated by a birth process, pure birth process, just particles splitting at random times. When you get the tree, you then start out some characters at some value. I think here we started right at the threshold. We set the threshold to 0. Obviously, the liability scale is arbitrary, and it's natural to put the threshold to 0 just because that's the most special place on the scale. But you could change it, make the threshold be at 3.14159, and the model would still be the same. Now you can then take Brownian motion characters and evolve them up the tree, and when you get to the tip, you just threshold them. Just get a 0 or 1 out of each one, and there you've got a simulation. And this outcome came from an actual simulation. So what I want to show you is doing a test this a very small scale simulation, the one I used in the 2012 paper on this. And what I assumed, I said, well, let's have some characters, or 3 characters. Let's have some of them be positively correlated, some of them be negative, and some of them uncorrelated. So here is the true covariance matrix of evolutionary changes. The variances of the 3 characters are different. There's a positive correlation between the first 2, a negative correlation between 2 and 3, and zero correlation, zero covariance, and therefore zero correlation between 1 and 3. So I thought that's okay. And now here is a quick test of to what extent is our Markov chain Monte Carlo run long enough. And what I did was to have a tree, evolve a set of characters up that tree, take that same set of characters and that same tree, and do an MCMC run estimating the covariance matrix, and then do another MCMC run starting with a different random number and estimate the same things. And if you look at them, it's reassuring. I think that this is the wrong covariance. Okay, it's supposed to be 1, but it's 1.7. Why? Well, you know, the characters, there's only 50 species on this tree. There's some noise in the estimation. But when we run the 2 times, we get something close to that 1.7 both times. Okay, the 1.7 is not just noise from the random sampling in the MCMC. It's the result of the randomness of the character evolution. And if you go through these, you'll see they're pretty close. They're good enough. This is from, you know, something like a 1-hour run with, I don't know, 100,000 steps or maybe 1 million steps. I can't really recall. Now, if you do that for a whole bunch of trees, for 50 different trees, and for obscure reasons, I did it twice on each tree. So we got 100 samples, all simulated with the same covariance matrix. 50 different evolutionary outcomes with that covariance matrix. And we then say, what do we get? What I have here is the estimates of the correlations between the characters. For obscure reasons, I preferred that to getting the covariances. The correlations, you know, the covariances scaled by the standard deviations are a little easier to look at. They go from -1 to +1. From those covariances, I can calculate that the correlation we should get between character 1 and character 2 is plus 0.53. The correlation we should get between character 2 and character 3 it's actually minus 0.5145. I left off the minus sign, but you can look on the scale here and see. And here it's supposed to be 0. So we look and we see, yes, we appear to get a cloud of results that centers around the true covariance. That's the good news. The bad news is it's quite uncertain. It's quite variable. And I'll come back to that in a moment. It's a little depressing in that respect. So it's sort of doing the right thing and sort of estimating the right covariances. Now, then I was getting ready to publish the paper, and I thought, you know, it's a pretty good method. We can use it on discrete characters. And then it occurred to me, you know, sometimes you have data sets with both discrete and continuous characters. Gee, won't we have to develop some terribly more complicated model to deal with that? That's going to be hard. Wait a second, that's easy. Because it suddenly occurred to me, actually, what is a continuous character? Well, it's just like your liability, only you observed it. So what do you do with a continuous character? You can put it right into this framework. You no longer worry about the 1s and 0s. You just observe the liabilities, and you keep the tip at that value. Don't let it change. So all it is running the same algorithm, but just put an if statement in that says, oh, that one's a continuous character. We know its value. Don't change it. And only change the interior node values. And then you can do the analysis. So you can run a set of data that has some discrete and some continuous characters. as long as you tell the program which ones don't change the tip values on, and all the rest of the machinery worked. So I even said, well, you know, we could run all continuous characters. Well, of course, that's a case for which we have fast phylogenetically squares methods or contrast methods. So I ran it through contrast, I got numbers, and then I ran the MCMC on just continuous characters. And yes, you do get basically the same covariances with a little bit of noise, but you get them. So it was reassuring. So that is, assume the tip liabilities are observed and don't change. You could estimate things. Noisier version of what we can do already if there are just continuous characters, but you can do both discrete and continuous characters. That was what they said there. And here is a test of that with the same parameters, the same trees, the same data, except that character 2 was assumed to be discrete and the other 2 were assumed to be continuous. And what you get is, now this is not correlations, this is covariances. That's why the distributions are suddenly much narrower. They really are as noisy as the other ones. Here is between character 1 and character, the variance of character 1, the variance of character 3. I think another reason that they're narrow is with continuous characters you get more information. You have a better, you don't just get which side of the threshold you're on. Here's between character 1 and character 2. And yes, it's positive. It's supposed to be positive. Here is the true covariance at the red triangle. Here's between 1 and 3, that's 0. Those are now continuous characters. Here's between 2 and 3, and that's supposed to be here and varies around that, and so on. So it looks like it's working. So we have a machinery now for 0, 1 characters and for continuous characters and mixed with each other. This, there are data sets out there. This one is a little bit, you know, a little dubious for a couple of reasons. One is you're not going to have a molecular phylogeny for it. These are fossil elephants. The only ones which are alive are the 2 species, or now 3 species of Elephas. Mammoth was alive till recently. This is a fossil data. I just want to show you a 0-1 data set by Pascal Tassi and Pierre Darlou. And there it is, and yeah, you could do things on this. You don't really have a molecular phylogeny for these, however. The other thing I should say is there are question marks in there. They mean we don't know if it's state 0 or state 1. What do you do in that case? In that case, you just tell the tip, don't worry about the threshold, change wherever you want, and let it wander around, because, you know, anywhere it goes is okay with the data set, fits the data set. So unknown, you know, missing data is very easy to accommodate in this. Okay, so there are things in the future that could integrate with this. We could imagine somehow QTLs in getting involved. I don't really have a scheme for that, and I think that's for the future. Models in which there are loci that we don't know, but we can infer their existence, that have big effects in data sets, and some residual stuff that will evolve according to the continuous character model. You would see, by the way, if you could get to QTLs, that you could see them change across species, including some QTLs causing variation within some species, but then not causing variation within other species. In principle, the interesting thing when you start thinking about QTLs is there are ways that you could actually answer the question that we now think we cannot answer in principle when we see change in 2 characters that are genetically correlated with each other. Which one is under selection? We all know that that's kind of not available to us, but with QTLs there are ways you could look at the data set that would tell you which one's under selection. Because basically, if there are QTLs contributing to one character some contributing to both and some contributing to the other character, which ones are the ones that changed. I will finish up there, but let me just show you the literature. Here's Sewell Wright's 1934 paper. There were two papers, one the methods and one the theory. They're adjacent papers in genetics. Genetics of that age is, by the way, freely available online at genetics.org or through your favorite library. Douglas Falconer in 1965 used the model to model susceptibility to diabetes, to juvenile onset, type 1 diabetes, and look at incidences in relatives of different degree. That was a major republicizing of Wright's model. And then animal breeders have picked up on this already. Dan Gianola in Wisconsin and his co-workers, Sorensen, people in Denmark, have used the threshold model in quantitative genetic, fitting quantitative genetic models to pedigree data, doing all the machinery. So I'm not being as novel as you might think. There's material starting with this paper in Journal of Animal Science. They have a big book, and there's a whole chapter on threshold models and how you do them. And here is Paul Lewis's paper in Systematic Biology on MK, Mark Pagel's paper. It's at the very end of 1994, so sometimes it gets cited as 1994 and sometimes as 1995. Me mentioning that you could do this, me giving more details. This is a preliminary version of the final paper, which is 2012. This came out because I was contributing to a Festschrift volume for Bill Hill, and this was something that was in the works but wasn't working yet. So this is kind of preliminary and also describes things you could do that turned out I didn't do. That later on got done a different way. There's some material mentioning it in my book. So that's all that I have for the moment. You'll be doing in a moment an exercise on this, but let's have some questions. And I should say, the use of the threshold model to model discrete states in statistical work on phylogenies is gaining traction. Originally, MK model was getting a certain amount of use. There was a lot of parsimony work, and I put this forward, but it has been gratifying that more and more people are slowly saying, maybe that's a better way to model discrete characters. Questions? One natural question people ask is, what about 3 states? And the answer is, and they say, couldn't you just put 2 thresholds? And you can in principle. But then, although the first threshold can be put anywhere, the question of how many standard deviations away the second threshold is becomes something you have to estimate. And it's worse than that. Because who says they're on one scale? It could be a 2-dimensional scale with a threshold for character 1 for going between state 0 and the others, and then the others having a threshold maybe going this way. So you then have a somewhat uncertain issue of how many liability characters should we have and what kind of regions should we carve out for each state. And there's a lot of work ahead of us to do to model that. I would think for meristic characters you probably should keep them on one threshold, but you have to at least know that you sometimes might want to go to more than one dimension to do higher states. Brian?
I think Liam has multiple states in PhiTools.
Probably on a single scale.
No. Is Liam still here? Yeah.
Do you have multiple states in your threshold? I can't hear you, but we need to give you the mic.
For a single character only. So for reconstructing ancestral states for a single character.
Well, but you did that by putting multiple thresholds on one scale.
Multiple thresholds along one scale.
How'd you decide where to put the second threshold?
I sampled them.
You said, okay, so he does it by MCMC.
Yeah, by MCMC.
That's a cute idea, but put in a reversible jump machinery and go into sometimes into higher dimensions.
Yeah, yeah. Or I mean, the other issue is that you have to specify a priori the ordering, so you could
Yeah.
sample the, so
Yeah.
yeah. Alternatively, you could
You don't
you
want-
could use you could
Yeah. You don't want state 8 to be in between states 2 and state 3, state 2 and 3. Yeah. Sorry, other questions? Susan.
Thanks. How well would this work? Is this working? How well would this work if you had relatively few genes controlling your trait, so that your distribution was your liability, the unknown liability, was lumpy?
A, it wouldn't work. B, it would work. I think if you've got 2 or 3 genes, probably 3, I think given the amount of noise in the whole thing, I think a normal distribution model would actually fit pretty well. If you're down to 1 locus, not so sure. 2 is marginal. But all this is based on my seat of the pants feel and prejudice. Somebody needs to go out and test it. And by the way, there's really nothing, I kept using the word morphological, but there's nothing here that says you can't do this for behavioral traits. You just assume there's some continuous propensity to give a bird having a propensity, the liability would be a propensity to issue a call or something like that, and that would work. Yes.
So if I understood well, in your when you were like validating the approach, you did like a lot of tree sample from the same covariance matrix, and then you got the distributions, right? What's this?
Well, I mean, in the simulation, I could afford to have 50 trees and run evolution 2 times on each tree. In real life, we've got 1 tree and 1 run. So you can't do exactly what I did. You would just get a number out, and the question would be how accurate that was. You could rerun the MCMC, but you don't but that doesn't measure the noise that comes from the stochasticity of evolution itself. It just tests the stochasticity of your sampling in your MCMC run, which is something you can redo.
And is that like a, because I think that could be useful, right? I mean, if you get like some distributions at least at the tips, and maybe even at the internal nodes, you will at least test how consistent the answer of that problem is.
I think you could do something like, it's been mentioned here, the difference between a non-parametric bootstrap and a parametric bootstrap. But parametric bootstrap, which was first described in the literature also by Bradley Efron, guy who invented non-parametric bootstrapping, is you're basically running a simulation like mine. You choose the tree, you use the actual tree you have, and you use your parameter estimates, and then you simulate under those, and then you can do multiple of those simulations and see how noisy your outcome is. A parametric bootstrap would be a good way to get some idea of the noise in these covariance inferences.
Okay.
There was 2 other people. You had the mic first, yeah. Susan has another question too.
Yeah, so this is all fairly new to me. Maybe you said this and I didn't pick up on it. Would it be possible, if you understood the kind of mechanistic basis of the trait, to actually measure or quantify a liability?
Yes, but we don't have many cases in which we can do that. We'd ideally like to get far enough along that we could measure the mechanistic basis, and then perhaps the natural selection might be on the 0-1 trait, but in terms of our data analysis, we would just go to that and use it as a continuous trait, and we would. I think there'd be some further rethinking to do if its fitness effect was through the discrete trait, but that would be much better, and you would have a much better hand on the liabilities if you could actually observe them. I don't know of a case, but there might be some out there. Yeah, you're next.
I may have answered my own question in my head, but I mean, I was just going to ask, what do you do about if there may be some underlying unknown structure to the liability that you don't know? For example, if you have multiple subpopulations within your group that have a different threshold, which, like for the classic diabetes example, I mean, we know that different human populations differ in their propensity for
Yep.
diabetes, but I guess you would
Yeah,
just
you
test
could. I
ahead
mean,
of
there
time,
there
or- yeah.
there are many complications you could have with geographic variation, and like measuring geographic variation on ordinary continuous characters, most of that territory has not yet been explored. There are just a few beginning approaches to that, and yes, it would involve a lot of extra machinery and rethinking and new modeling, but it does in the straight continuous characters too. All of that needs to be further developed. I should say that I haven't got it in my program yet, but it is very clear how one could use this. I can sketch out in my mind how to do the program where we have within species variation, so we're collecting 0-1 information on individuals within a species, and we want to have, in addition to the evolutionary covariance matrix, phenotypic covariance matrix within species, which would be parallel to what I did in 2012 and what Mike Lynch did in 1991. And we could estimate both the within covariant species covariance and the between in a unified way. And there's no ambiguity about how to do it. I know how to do it. The first step is hire a programmer, and or have the time to program myself, and I haven't got that done. Sophia?
It's not a real question, but I think we've got a bit.
Is any question in this course a real question? That's
Okay.
the question.
It's just a call for some, because I think we got a bit confused here about the simulation. So so if you could go over that, it's slide 26 briefly with
Right,
the
it's back-
true.
it's-
So we have the.
Wait, I've got the... You mean this one or
No,
the previous one?
previous.
Yeah.
Oh yeah, here. So the true covariance matrix. This is the true covariance
This is the truth,
between the
because
traits.
I am the deity in this
Yeah.
simulation. Yeah.
So this is for the traits, and then for the simulated data set, is that after? Because you're using 50.
It will look, each simulated data set, well, it'll have 3 columns instead of 2.
Mm-hmm.
It'll have 3 characters, and it'll just be zeros and ones for each of them, for each tip. And we're assuming, as we have with pretty much everything we've done here, that you're not estimating the tree. The tree is given to you by people doing vast amounts of molecular work.
Mm-hmm. And in this case, we have 50 different trees.
Have 50 different
trees.
Well, I have in 50 different tips on a tree and 50 different trees.
Right.
And they were simulated by, pure birth processes ending up with 50 tips.
Right. So, but then, so our true matrix, it's what we have at the root.
Our true
that-
covariance matrix is the evolutionary covariances of change of the continuous liabilities along this tree. So you imagine yourself doing covarying Brownian motion, covarying according to that covariance matrix, not just at the root, but through the whole tree, on every branch. And when it ends up with liabilities up at the tips, and then you just threshold them, decide which ones are positive and which ones are negative, and make your zeros and ones.
Okay. But then for each tip, we have this, each tip is a species which has these 3 traits
Right.
and has the covariance matrix, but for each run here, we get on the next slide, we get a single result. So is that?
Yeah, there's a result. There's a result from one run.
But this is the result across the whole, because this
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 is the
matrix,
result analyzing the full set of species on the tree,
right? Okay.
doing all this moving of liabilities around and summarizing their changes
Mhm.
in an estimate of the covariances. Here's the result.
So this is among species. That's what I'm asking. So
This
you get
is
a
yeah.
single entry, a single data point for this variances and covariances is a mean trait value for a species. Is that
the
correct?
data you feed
The-
in is 50. No, you don't feed in the mean you don't feed in the mean liabilities. You don't see them. You know they're in your simulation, but you don't see them. You see the zeros and ones. You feed in a 50 by 3
Mhm.
table of zeros and ones.
Exactly.
You give it a tree.
Right. Right.
It then sets up liabilities and moves them all over the place and
Mhm.
does things with them and ends up with this as its estimate of the covariance matrix for of changes in those liabilities.
Yeah. So in other words, we don't have within species variation in a trait.
In
A
this
species
model,
has
no,
a-
we
a
don't.
tr-
We assume we magically
Right.
are able to observe the species mean without error,
Mhm.
or observe without error whether the species. It really, in a sense, it's the modal value. It's the value at the 50% point of the distribution. It's basically whether more than 50% of individuals in the species have a 1 or a 0.
Right. Right.
That's really, and you've forgotten about the within species variation. In the future, we will be able to do that.
Right. Okay. And then you mentioned it, but so we have this bias in, I think it's a bias, right, in the estimates, because between the true covariance matrix and the real ones.
And that's
So-
because this is the result of one outcome of evolution.
Mm-hmm.
And that outcome might have made it look as if there was a higher correlation than the real covariance than there really was. So we can see here that on repeated MCMC runs, we're continuously getting values up near 1.7 when we really should get a value, there we go, a value 1.
Mm-hmm. And that's the case.
That's because of the noise of the
Yeah.
of the data outcome in evolution.
Mm-hmm. Mm-hmm. Okay. Okay.
Right? Yeah,
Thanks.
no, it's
Good.
helpful to go back over that. Yeah.
I see here that it's it could be more than, or at least two. Well, you're doing 2 potential nucleotides right here, but what about something that could have 4 variations or something like that?
Yeah, you're thinking of traits that would be instead of 0, 1, they would
Yeah.
be, they could be meristic traits in order, like how many scales, 0, 1, 2, 3, 4,
Mm-hmm.
or it could be just discrete alternatives. Does this kind of dance versus does that kind of dance versus does another kind of dance, where you can't easily put them on a scale. But what I was saying was basically, for 2 alternatives, we got the machinery. For 3, it's hard to know what to do, but you might put another threshold on the same scale, or you might go on to a new scale, and Liam had got had got one of those cases actually running in his implementation. So there's work to do to go ahead to multiple states. It has the very interesting possibility that with enough data, you could make interesting inferences about, is this display in between, is this song in between this song and that song, or is it somewhere else? I should say the one thing, the one message I wanted to get to that I didn't put up a slide for, but the really depressing thing here is, yeah, I've made all this work, but I've made it work with a lot of noise, and there were 50 species there. So the one thing you're going to say is, you might say, well, you could get more species, that would make this better, but you may run out of species, or run out of species you can get specimens of. Doing more MCMC runs does not make these distributions narrow, or doing longer MCMC runs removes a little bit of variance that comes from the MCMC sampling, but it doesn't remove most of the spread. The result is here, look, we got it about the right number, but that's on average over many cases. In practice, it was spread, the correlation was just somewhere between 0 and 1. That's pretty noisy. Look at this one, goes almost from -1 to +1. This one also spread very widely. The conclusion is, yes, you can get ideas about the covariances, but they're very noisy. And this is part of the fly on the wall problem, that the data available to us and the species available to us have limitations in how well we can estimate things like this. And if you're not ready for that, you're in for a lifetime of disappointments.